So here we are with genetics, and I've tried to put things together in the order of priority that you indicated on the form, those of you who filled that in. So I'll run through those. I'm going to start with, it's kind of hard to do anything with genetics without definitions, so we're going to start off with a few of those. And apologies, I might not be able to see the chat very easily while we're doing this, but I will try and flick back over and answer questions that I can see. And I'll put the slides up linked in the video as well. They're a little bit disjointed, just because it's my thought process rather than anybody else's, but hopefully they'll be able to help you. All right, so starting off with genetics then, here we've got the kind of classic picture of a cell with a nucleus, a chromosome, a gene and DNA. And for an achieved level response, you're probably going to need to define some or all of those terms. So the cell is the basic unit of life, and inside most cells there is a nucleus, and that nucleus contains genetic information in the form of chromosomes. So humans have 46 chromosomes in most of their cells, and those are these X-shaped structures here. So those chromosomes contain or are made of the chemical called DNA, deoxyribonucleic acid, and that chemical is really just a series of nucleotides. And when we say nucleotides, we're talking about the A's, T's, G's and C's, and we can see those letters on the DNA molecule here. Now the gene is often the kind of harder part to define, and a gene is just a region on that chromosome. So any chromosome will have lots and lots of genes, and each gene will be in a particular place along the length of that chromosome. Okay, so I think I've covered the main definitions there. In terms of how to form an answer for this one, I would probably do you could do each term individually, or what I've just done, which is kind of take you through a story of, if we start off with the cell and go to the nucleus, and then what are the structures inside the nucleus, and then I went to the chemical molecule, DNA, and then defined a gene as being a region on that chromosome molecule, a certain length of DNA in a particular place. Now I've thrown this one in here. I'm not sure that I put this on the form, but it's also really useful to be able to define a mutation. And so because we've just been talking about those nucleotides, that genetic code, it makes sense to jump onto mutations. So a mutation, and this statement you probably just want to memorize. It's a random, unpredictable, irreversible change in the genetic code of an organism. So it's a permanent change in the sequence of A's, T's, G's and C's, those nucleotides in the DNA of an organism. And to help you form your answer, I would suggest that you link each term that you've been asked to. So you can see in the question here, there's words in bold. You want to really explicitly state those, and then define each one of them as you go through your answer. So to answer this question, explain how mutation can give adults the ability to digest milk. So a change in the genetic code of our DNA, that's a mutation, can create a different version of a gene. So we call different versions of a gene an allele, and that allele may code for a new protein. So every gene codes for one protein, and so a different version of a gene can code for a different protein. And in the case of humans, this mutation that happened, I think possibly in the region of 15 to 20,000 years ago. I may be wrong on that, but that's the ballpark figure in my head. happen predominantly in North Africa and Europe, and those populations develop this ability to digest milk past the age of about 3. That's just background information, you don't really need to know that. But that change in the gene, that mutation, creating a new allele, changed the phenotype, so what was actually expressed for those humans. And that meant that beyond the age of 3, those humans with that mutation could digest lactose, the sugar that's in milk. And in order for that mutation to be passed on to the subsequent generation, the mutation's got to happen in the gamete, the sex cell of one of the parents, in order to be passed on to their offspring, or in the cell that forms the gamete. Okay, so I think that allows us to move on to the next big topic that a lot of people wanted some help with, which is meiosis. So here's a bit of an image of meiosis. I've tried to pick one that's got a bit of color, different color for the different chromosomes. There's a few terms in there that you don't desperately need to know. So I wouldn't worry too much about the centrosomes or the spindle. What we need to know about meiosis is that it happens in our sex cells, and the end result of meiosis is the production of gametes. So in humans, that's the sperm or the egg. In plants, that would be the ova or the pollen. One cycle of meiosis would begin with 1 cell and would end with 4 genetically unique cells. And in order to achieve that, the genetic information has to halve. And so if we were dealing with humans here, we would have 46 in the initial cell, and then we would end up with 23 in each of these 4 cells at the end of the process. If you're struggling to remember the distinction between mitosis and meiosis, I try to remember that mitosis happens in mitose, and so that's your body cells, not your sex cells. And therefore, if you can remember that, then you know that meiosis is the one that happens in your sex cells, or leads to the production of your sex cells. There are 3 things that happen in meiosis that you might be asked to talk about, and those are highlighted with arrows on the diagram. So we've got this thing called independent assortment, and I'll go through and talk about that in a bit more detail in a moment. We've got crossing over, and we've got segregation. So segregation, again, I'll talk about a bit more, but it's the separating out of chromosomes to different ends of the cell. And so in this case here, we can see the green and the blue chromosomes end up in this cell, which then goes through another stage of division to produce the 2 of the final 4 cells. And on the lower part of that diagram, we've got the red and the yellow chromosome, and they end up being separated out into this cell, which goes on to produce these 2 of the 4 gametes. So the reason that you might have to know about these 3 different processes are that you could be asked around what happens in meiosis that leads to variation. So what can happen in meiosis that leads to variation? And these 3 different things are aspects that you could talk about that lead to variation in the gametes, which leads to differences in offspring. Okay, so having a bit of a closer look at independent assortment, and I apologize, I've changed the direction, the orientation of the diagram here, but hopefully you've seen enough of these to get your head around that. Independent assortment I find is one that's probably easier to talk about, because all you need to do is identify that when the chromosomes line up along the equator of the cell, they pair up in homologous pairs. And what that means is that the same length of chromosomes pair up with each other. Now, in this individual, we might suggest that the red chromosomes came from their mother and the blue ones came from their father. And so in each of us, we've got one chromosome that came from one chromosome of each of our chromosome pairs that came from our father. So we've effectively got 23 from dad and 23 from mum. And each of those chromosomes that we got from dad is a slightly different size, and each of the ones from mum is a slightly different size as well. But one of the ones from mum is the same size as one of the ones from dad, and that's what we mean by homologous chromosomes. And what we find on those homologous chromosomes is that if there's a gene at the tip of one of the chromosomes, the one from mum for example, then that corresponds to the same region on the chromosome from dad. And so in the picture here we can see that as the little yellow band on the chromosome, and we're saying that this region of the chromosome is one gene, and so we have two copies of a gene. We've got one from mum, one from dad. Those could be the same or they could be different. If they're different, we talk about them as being alleles, i.e. different versions of a gene. So in independent assortment, those chromosomes align themselves along the equator of the cell, and so we've got the two homologous, the larger homologous ones paired up here, and then two other homologous chromosomes. These ones are a bit smaller, paired up in the bottom here, and that is really independent assortment. So it's just those homologous chromosomes lining up. Now it could be that you have all of dad's forming on the left hand side and all of mum's on the right. It's quite unlikely. What's more likely to happen is that you're going to get, this one's dad's on the left and this is one of mum's on the left. And remembering this is a simplified diagram for humans, we're dealing with 23 pairs of these chromosomes rather than just 2 pairs of chromosomes. And so independent assortment just means that, dads could go on the left or dads could go on the right. So in these lower two, we can see that dad's blue chromosome's on the left and mum's is on the right. Whereas on possibility 2, we've got mum's on the left and dad's chromosome on the right. So that's independent assortment. Okay, the lining up of those homologous chromosomes independently of every other pair. Then we have segregation. Segregation, I find, is probably a little bit harder for students to explain in an answer, so you might not want to try and tackle that one. But segregation is really just the splitting of the cell. So once we've got the homologous chromosomes lined up in a random or independent assortment, then when those cells separate, those chromosomes separate to opposite ends of the cell, we get two new cells being formed, and the mixture of those chromosomes is determined by independent assortment. So in possibility 1, we've got dad's chromosomes have formed into one of the daughter cells, and mum's have formed in the other. In possibility 2, we can see because the stage of independent assortment was different, then when segregation happens and those chromosomes are pulled to either end of the cell, then we've got one of dad's chromosomes and one of mum's chromosomes in each of those cells. So that's segregation. And remember that that's going to lead to, any cycle of meiosis is going to lead to 4 gametes, which have half the genetic number. You can use the term haploid, H-A-P-L-O-I-D, to mean half the genetic number. Okay, so the last one for meiosis is crossing over. And in crossing over, when those homologous chromosomes are lined up on the equator of the cell, we sometimes get one of the legs, if you like, in this diagram, crossing over with the leg from the other chromosome. And when that happens, we can actually get a swapping over of genetic information. And we get what we call recombinant chromatids. And so the recombinant chromatid, if I follow it with my cursor here, we've got a little blue chromatid line here coming down to, sorry, I should probably have followed this one down, my bad. down to here, and so it has recombined. So the blue chromatid has recombined with some of the genetic information from the red chromatid. Whereas this chromatid over here on the left, if we follow that up, that's just blue all the way, so it is a non-recombinant chromatid. It hasn't recombined through crossing over. Terms like recombinant and non-recombinant, you don't need to know. You just need to be able to describe what is going on in crossing over, that swapping of genetic information, which increases the variation in the gametes. And I can show you that, I think, if we go back into this diagram. Oh, no, my bad, we don't have it in that one. So I just have to go from this diagram here, and each of those chromatids being separated out into one of the 4 gametes. We might have, one gamete that just had the blue chromatid, one that had just the red, and then 2 of them would have these recombined chromatids. So the one red and blue, and the one that's blue and red. And that means that if a gamete containing this chromatid went on to fertilize an egg, then it would have a new combination of genes that hadn't existed in either parent. If we stick with blue being from dad and red being from mum, then this one would have a length of the blue chromatid from dad and a bit of the red, which was from mum. Now that didn't exist in dad, because dad was just blue all the way, and mum was just red all the way. So we've got variation there, a recombining of the alleles from the genetic information we received from our parents. Okay, I'm just going to flick back over to chat, see if there's any questions that I need to answer on that one. So we've got a good question there. Is segregation random fertilization? No, and I'm really pleased you asked that. So fertilization is where one sperm fertilizes one egg. The average male ejaculation in humans produces about 400 million sperm. And so the random fertilization means that it's only going to be one of those sperm that fertilizes one of the females, normally about 400 eggs. But only one of those eggs is released in any menstrual cycle. So we get the egg, and that's released at random, if you like. It's one of the 400 that female human had when she was born, and that matured throughout her life. And one of 400 million sperm is going to meet that egg, and that is random fertilization. So it's that idea that we've produced a whole load of different gametes, and it could be any one of the male's gametes fertilizing any one of the female's gametes. So that's what we mean by random fertilization. So I had a good question there about is segregation, I think was the question, the movement of homologous pairs to the equator of the cell. Yeah, you're almost right there, but the homologous pairs lining up on the equator of the cell, if I go back to this diagram, I would identify the equator of the cell as being vertically down the middle here. And so that's independent assortment. Segregation is when those chromosomes get pulled to opposite ends of the cell. And so the blue chromosomes in this example are going to get pulled to this end of the cell, where my cursor is now. And the red chromosomes are going to get pulled to this end of the cell. So independent assortment is them lining up along the vertical line. And they can do that in different orders. Of blue or red being on the left, and then segregation is where they are pulled apart to opposite ends of the cell, and then the cell splits, and that gives us these different cells as after the first stage of division in meiosis. Yeah, I hope that was clear. I'm not entirely sure if it was, so if it wasn't, ask for clarification. Any others? All right, if I see any more questions come up, I'll try and deal with those as we go through. I think we're doing quite well for time, so hopefully that that aspect of meiosis makes sense to you. I would expect a good question on meiosis where you had to explain crossing over or independent assortment or segregation as probably being a merit or an excellence level answer. Without knowing, I've seen a question about is my real exam going to be harder than the mocks? I don't know which mock exam you've sat, I'm really sorry, so I can't answer that question, but often as teachers we might give you a slightly harder one for your mock exam to help prepare you for the external. What I would suggest you do is just try and do as many NCEA papers as you can to prepare you for the external on Friday. Okay, so I'm going to move on now to that idea of variation, and this is quite a common question in the genetics paper. I quite like this one because it asks us to define sexual reproduction, to explain how one important process in sexual reproduction helps to produce variation in offspring, and hopefully that question is really making you think of crossing over or independent assortment or segregation. And then finally, and this is a real excellence part here, how does variation through sexual reproduction benefit the Venus flytrap plant population over generations? I think we see that question quite a lot in exams, so it's really good to be able to prepare for that one. All right, so I'm not worrying about the specific context of the Venus flytraps here. I'm just really tackling each bullet point as I go through. And that's a good strategy. You want to be ticking off each bullet point to make sure that you have answered every part of the question you've been given. So, sexual reproduction is the creation of a new organism through fertilization between a male and female gamete. So dropping in those keywords there that demonstrate our understanding: fertilization, gamete, new organism. Each gamete contains half of the genetic information of that parent. And these recombine in the zygote to give the full genetic number. I'm not going to go through explaining crossing over or independent assortment again for this, so I've just written in that I would explain those. If you need more time to digest what I've gone through there, then the video will be up on YouTube after this, and so you'll be able to play that back and hopefully make a bit more sense of what might be a very quick and rapid giving of information. And then the last bullet point in here, explain how variation as a result of sexual reproduction can benefit the Venus flytrap plant population over generations. Anything that's relating to over generations should really make you think of natural selection. That idea that the best adapted to any set of environmental conditions has a slightly higher chance of surviving to maturity and reproducing, and therefore passing on, whichever alleles it was, whichever versions of genes it was that gave that organism an advantage in the environment. So what I've done here is put together a sort of an answer. It might even have been my colleague Michael that put this one together, so kudos to Michael if he did this one. Variation improves the chance for some members of a population to survive in changing environmental conditions. So it's really key that you realize that there will be over time different environmental conditions, and those different environmental conditions will put pressures on a population. Members of the population whose alleles are better suited to any given condition will be more likely to survive and reproduce, passing on more of those successful alleles to subsequent generations. It's quite a bit to take in there, so I might just take a moment to go back to the milk example, the ability for humans to digest milk. And I think 65% of the world population doesn't have the allele to digest milk after the age of about 3. But in populations, 10 to 15,000 years ago, particularly in Northern Europe and Africa, if there was, famine or drought or a limitation on food supplies, if some members of a population could supplement their diet with milk, then they had a better chance of surviving and reproducing. And it might only have been a tiny, tiny increase in your chance to survive, but over time, that comes through, and that means that those individuals do pass on more of their alleles to subsequent generations, and that's the idea of natural selection. And so the last part of that just says that sudden changes in the environment can be detrimental, that's like a bad effect, and in some cases lethal. But if we have variety in a population, then there's a good chance that some members of a population will survive, even as others die. A good contrast to that might be, if organisms produced through asexual reproduction, then going through asexual reproduction means that every organism within that population is genetically identical. And that means that if that was the case in humans in Northern Europe 15,000 years ago, then if none of them had that lactose, that allele that allowed them to digest milk, then it might be that the population as a whole couldn't survive, and so the whole population might have ended up being wiped out. But with sexual reproduction, you've got variation, and that allows some members of the population normally to survive changing conditions and then to pass on their genes. Pedigree charts. I'd definitely expect to see a Punnett square question. So I've got a Punnett square here that's being pulled from a pedigree chart. And I'm just going to quickly go over this one because I think most of you felt you were pretty confident with your Punnett squares. So the Punnett square is this little thing down here. And from reading the question, you'll normally be given a letter that represents a gene. And so in this case, our gene is the letter T, and the two different varieties of that is little t, which we're told is cystic fibrosis in the question, that's recessive, and capital T, which is the unaffected allele. We know that the capital one is dominant, the lowercase one is recessive. And so if we have to construct a Punnett square, then in this case, if we were asked to demonstrate the cross between member 1 and 2, then I might have number 1 up here and number 2 on the side here. Doesn't matter which way around you put them. So what we're doing here is we're picking out from the question. We know that these two individuals have had offspring that are affected, meaning that they must both be little t as their genotype, which is homozygous recessive, is a fancy term for saying that. So that tells us that each of these parents must have a little t, and in order for them to be unaffected, they must also have the dominant unaffected allele. So both of these guys, the mum and dad, are both heterozygous, which means they have one big T, one little t. And so I would write the genotype for each parent in here, and then I would separate that genotype into the two different alleles. So I've got a big T here, little t, big T, little t. And then I'm just filling that Punnett square. Each of those boxes is taken by combining the alleles that are in line with it. So this big T gets copied across into each of these boxes. I might actually, what I'll do is I'll just delete those and go through it step by step. So if I focus on this allele, then I'm going to put a capital T in here. And going with this allele still, I'm going to put a capital T in this part of the Punnett square. If I then go down here, and I'm looking at the recessive allele, I'm going to put that into this box and into this box. Okay, I've now finished dealing with this parent, so I'm going to go up here and I'm going to take the big T. It's by convention we write the dominant letter first if it's heterozygous, and so I'm going to put the big T in here, and I'm also going to copy it across into this cell down here. So I might end up writing that, and that's not entirely wrong, but it doesn't fit with the way we normally do it. So I'm going to turn that around into big T, little t. I'm now looking at this allele, and I'm going to take that little t and I'm going to copy it into this cell, and I'm going to copy that same little t allele into this cell. And so that gives us our completed Punnett square, and we might be asked to look at the phenotype or phenotypic ratio or the genotype ratio from that. If I was asked about the phenotypic ratio, that's what is expressed in the individuals, then from this Punnett square, I would expect to see this guy's unaffected, this individual's unaffected, and this individual's unaffected, whereas this individual is. That means that there's a 3 to 1 chance of being affected, or of being 3 unaffected to 1 affected or sufferer of cystic fibrosis. That could be expressed as a percentage as 75% unaffected to 25% affected. And we notice that while we predict that ratio, that 3 to 1, we can see in reality that wasn't the case. And so that comes back to random fertilization. While we can predict what is expected to happen, especially in cases where there aren't many offspring, the actual ratio could be very different, because this individual was a product of random fertilization, one random sperm with one random egg, and it just so happened that that sperm carried the unaffected allele, and that the egg either carried the unaffected or affected. As long as one of the gametes carried the unaffected allele, then this individual number 3 is going to be unaffected. They could also be homozygous dominant, which would be big T, big T. Okay, so it'd be good to be able to discuss random fertilization as being the reason for the expected phenotypic ratio not matching the actual phenotypic ratio. Okay, so moving on to, this is more pedigree charts, and so we're really just looking down through here, identifying the genotypes of the following 4 individuals. So number 1, this is now a different condition, just to highlight. This is called photic sneezing. We've got capital A is dominant, and that means that you might sneeze under particular lighting conditions, and the recessive characteristic means that you're unaffected. So individual number 1 is, what have I got here, is affected. I'm just looking at my answers here, and I'm actually thinking those don't look quite right. Number 1 is, no, that's correct, yep. So number 1 is unaffected, and so to be unaffected they cannot have a capital A, so they must be homozygous recessive. Homozygous just meaning that both of the letters are the same. Number 2, this is an affected individual, an affected female in this case, and we can see from their offspring that they had a mixture of affected and unaffected. If the affected female was homozygous dominant, i.e. big A, then she could only pass on a capital A into every single one of her gametes, which means that every single one of her children would be affected. Because that's not the case, we know that she must have a recessive allele in there as well, and so she would have to have one capital A and one lowercase a. Number 11, so over here again, number 11 has offspring that are affected and unaffected, meaning that individual number 11 must have both the dominant allele in order to be affected by the condition, and the recessive allele if they can pass on a recessive allele to an offspring that's unaffected. We can also look at individuals 3 and 4 and use that to support our reasoning. Individual 3 is unaffected, which means that every one of individual 3's offspring must have a lowercase a, and so that means that number 11, being an offspring of number 3, must have a lowercase a, and so they must be heterozygous. Heterozygous meaning 2 different alleles, so 2 different letters. So that's what we're showing down here.
I think it just answered it.
So I've had a question there. What's the best way to answer questions of how dominant parents produce a recessive phenotype offspring? And I think that's what I've just gone through. So a parent in this case of number, we'll go back to number 11, shall we? So yeah, just to reiterate there, number 11 has produced, in this case, an unaffected female child. That unaffected female child must be homozygous recessive, little a, and so that tells us that number 11 in their gametes must have passed on in one of their gametes the lowercase a, and that gamete had formed individual 14. But because they are affected, we know that their other allele must be the dominant one in this condition, in this situation. Make sure you read the question carefully and just have carefully think about what each of those alleles, the dominant, means. And it might be useful to actually write down the genotypes on the diagram, and just to help that make sense to you. And then if you bump into an area you're not certain with, go back and double check them, just to make sure that what you've done is correct. Okay, I hope that helps. All right, so to finish off with genetics, it's just the actual and expected phenotypic ratio. So I think we've done all that already. So I'll just give you guys a moment if you want to fire through any questions. I'll give it another few moments, and then we'll start on acids and bases. So there's a good question there. How hard is it to get and achieved? It's normally, I would say it's not hard at all. The key information that you need in genetics for achieved is the definitions. And so what I would do is I would use whatever resource you've got, whether it's a workbook, Education Perfect, or visit the website called No Brain Too Small. I think that's actually got all of the definitions and the need to know information in there. I think it might be useful for us actually just to flick over to No Brain Too Small. For those of you who haven't seen that before, this is a really good website. It's created by some teachers. I know some of them work in Wanganui, and I'm always really grateful for this resource. So if we go into science and level 1 science,
maybe you could go through DNA replication or variation or try and find those.
and if I go into genetics, then there's a whole heap of resources here which are really good. And so the last minute reminder sheet and the glossary might be really useful for you. So if you were just trying to get achieved, and I wouldn't recommend that. I mean, I always want you to push for a higher level, and it shouldn't be too hard to get beyond achieved into the merit category at least. This information here gives you all of the definitions, so you can work on memorizing those and being able to use those, and that should help you get your achieved in the exam. And the last minute reminder sheet's really good because it kind of contains a summary of a lot of the information you need to know. You might want to go through that. I think it's just 2 pages. And then there's also collated questions. So if you know that you've been struggling with variation and survival, then maybe you go and visit those questions, and that's got all of the exam questions relating to that topic put together. And then further down we've got notes on the different aspects of genetics, and there's flashcards as well if that's something that might help you memorize some of the definitions.
So we've got pure breeding strain, and what about an excellence?
Okay, so could I go over pure breeding strain? only be able to do it very briefly, I'm afraid, just looking at the time. But a pure breeding strain is normally one that is homozygous for any characteristic. So that's either homozygous dominant or homozygous recessive. And normally in something that you might have learned about called a test cross or a back cross, you would normally pair an individual with an unknown genotype with another member of the species that you knew had the recessive characteristics, and so could only be homozygous recessive. And in that way you can determine whether the one that you were unsure of was homozygous dominant or heterozygous for that condition. So if it was heterozygous, you'd expect the offspring to be a mixture of the two phenotypes, whereas if all of the offspring had the dominant characteristic, then that individual could only have been homozygous dominant.
DNA replication is ×2 in protein synthesis. It's really up to date. It's really level 2, isn't it? I think protein synthesis is DNA replication. Oh yeah.
I wouldn't stress too much over protein synthesis and DNA replication. That's more level 2 biology concepts. What you really need to know for DNA replication is meiosis and mitosis, and so I'd probably look at both of those in a bit more depth, maybe using No Brain Too Small if you want a bit more on that. In terms of how to go about answering an excellence question, it's answering every one of the bullet points with the information that is requested. So your best bet to go through that, I often find, is practice. So I would be looking at past papers, having a go at those questions, and then checking the mark scheme and grading your own answer, seeing if it's on the right track, and then maybe in a different coloured pen I would go through your answer and add to it with the help of the mark scheme. Then pull up a different paper, have a go at those questions, and go through the same process. And over multiple goes of doing that, you're going to find that your answers develop, your understanding develops, and you know what to look for. I always reckon that if you do 5 past papers from previous years, then there's very little that's going to surprise you in the actual exam. Okay, so sorry if I've missed any questions that have come through. I'm going to move on now to acids and bases. So if you only had the genetics to cover, then thanks very much for coming along, and I hope it's been helpful to you. And if you're doing acids and bases, then that's what we're going to move on to now. Okay, so based on the feedback that you guys gave me in the form, rates of reaction was really high on the list. That was the biggest priority that most of you identified. So to go through that, I'm going to use a little tool that I've downloaded called AtomScope. But before we have a look at that, there's a few key areas you need to know about and you might be asked about with regards to rate of reaction. Temperature, surface area or particle size, concentration, and catalysts. And I can't recommend this video enough. There's a really great TED talk called How to Speed Up Chemical Reactions and Get a Date to a Dance, I think it is. But if you Google TED and How to Speed Up Chemical Reactions, I think it's less than 5 minutes, and it does a beautiful job of going through the different aspects of reactions, chemical reactions, and relating that to a kind of everyday scenario to help you understand it. So it's well worth watching that. Okay, so what I'm going to do now is I'm going to try and go through a few of those. All of our explanations for this, and this would normally be a kind of merit, high merit or excellence question if we're explaining it. Each of these should refer to collision theory, and collision theory centers around the idea of particles and the particles colliding with with sufficient energy and in the right orientation in order for a chemical reaction to take place. So So I'm just going to pause for a moment while I have a look at swapping over to atom scope.
This has a bit more and explain in detail, please.
Sorry, I've just realized that the thing that I wanted to do isn't in my browser. And so yeah, I'm not going to be able to share that with you, unfortunately. So what I will do is I'll mention this thing here called Atom Scope, and it's quite a cool little tool. You might want to download that and have a little play around with the chemical reactions in Atom Scope. Yeah, I don't think I'm going to be able to bring that up while we're running. Sorry about that. So I'll talk through each of these with reference to particles and the idea of collision theory. So with temperature, if we increase the temperature, then we increase the kinetic energy of the particles. So the idea is that we've got particles, and if the temperature is high, then it's got a lot of energy, so it's moving around rapidly. If it's low temperature, then it's moving around much more slowly. And that relates to how much energy those particles have, and we need them to have a certain amount of energy in order to react when they collide. So one that is at higher temperature has more kinetic energy, more moving energy, and it's more likely to react when it collides with another particle. Surface area. So if we can increase the surface area in a chemical reaction, we'd normally achieve this by grinding down the materials, or one of the reactants, one of the things that's going into the reaction. If we can grind it down, then we make the particle size smaller, and that means that we've effectively freed up more surface area, and the more surface area we've got, the greater the likelihood for an effective collision to take place, or a successful collision to take place, and the reaction to happen. A good example of that would be sugar cubes. So if you've got a sugar cube, big block of sugar, and you drop that into a cup of tea, then it's going to take longer to dissolve than if you pour in the same quantity of sugar, but in a granule form. And we could increase that rate of reaction further. So it's not a perfect analogy because it's not really a reaction happening there, but it works for the idea of surface area. If we were to grind down the sugar crystals to make them even finer, then that would increase the surface area even more. When we've got the cube, the sugar cube, then it's just the outermost molecules that are available for a reaction, and the reaction would have to happen for those outer molecules before it released or freed up the molecules that were on the inside of that shape. With concentration, if we had a certain number of particles, so if we say had 10 reactants of one type and 5 reactants of another type, then those particles would move around in a reaction, or in a container perhaps, and if they collided in the right orientation with sufficient energy, then the reaction would happen. So referring back to the idea of particles and collision theory. But in the case of increasing the concentration, what we're doing there is that in the same space, instead of having 10 and, what did I say, 5, let's say we had 10 and another 10, then the chance of those particles to collide with one another has increased. And if there's more collisions happening, then there's a higher chance of any one of those collisions having the right energy and orientation for a successful chemical reaction to occur. So concentration is about putting more particles of either reactant in the same space for the reaction to happen. And then finally we've got catalysts, and catalysts are another compound that we would introduce into the reaction, but a catalyst isn't changed by the reaction. What it does is it tends to hold one of the reactants in a particular position that helps the reaction take place, and then that makes it easier for the other reactant to collide and react. And so having a catalyst present increases the rate of reaction. You might expect to see a little graph with rates of reactions, and that could look very different depending on what's being measured. So it might be the mass of the reactants over time, and so you would expect the mass of the reactants or one of the reactants to decrease over time, whereas if it was the mass of a product, then you would expect that to increase over time. And being able to talk about the different shapes of graph is really useful. Generally, the steeper the line, the greater the rate of reaction, and that tends to happen at the start of a reaction when you've got the highest concentration of each of the reactants. Once the reaction's underway and particles have started to react, then the concentration of the reactants effectively decreases, and so then the rate of reaction slows down. Okay, hope that helps. Okay, a bit of need to know information for the acids and bases paper. You should know the names and formulas for these three acids. So hydrochloric acid, HCl, sulfuric acid, H2SO4, and nitric acid, HNO3. Okay, and that's the chemical formulas for those ones. You really want to go into the exam knowing those off the top of your head. You can work them out in the exam, but that's really just a waste of time. There's only 3 of them, so you should be able to, with a bit of practice, memorize those formulas. Any questions?
No, they've been saying collisions per units of time is important because it marks you down for excellence.
Yeah. It's a really good point in the chat, referring to the number of collisions per unit time. If you say the rate of collisions, then the word rate already means per unit time. But if you say the number of collisions, then it's important to include in that a reference to time. So the rate is the number of collisions per second, for example. Okay, and so with these acid formulas, we've got hydrogen in all of them, so you'd expect the hydrogen ion to be in any acid that you're going to be looking at in the exam paper. And normally when these compounds are dissolved in solution, then the ions separate out, and so the HCl might separate into H plus and Cl minus, and so it's the H plus, the hydrogen ion, that influences the behavior of the acid, makes it an acid, if you like. And I think we'll probably cover that in a little bit more detail in a moment. I noticed in the comments on the form, a few people had asked for the particular reactions, and I think there was one year where there was a question around baking soda, and people weren't really sure how they should have known what was being made, and so for that one you need to recognize that the formula for baking soda, I think it's a hydrogen carbonate, and so it behaves the same as an acid plus metal carbonate. So these 4 reaction types with acids are need to knows. So if we've got an acid plus a metal on its own, that's going to make a salt plus hydrogen gas. Acid plus metal hydroxide gives a salt plus water. Acid plus metal oxide also gives a salt plus water, and acid plus metal carbonate gives a salt and carbon dioxide and water. It's important to remember that whatever elements are present on this side, on the reactant side, have to also be present on the products side. So we can see that by having a carbon here, then there's going to have to be a carbon over this side. And the only time that happens is where you've got carbon dioxide being one of the products for the level 1 acids and bases paper.
So a question about rates of reactions. Do you need to define all of them for a higher grade? And
Good question about the rates of reaction. How many would you need to explain for the higher grade? It's really the key word there is explain to get the higher grade. And normally in an exam you would only be asked about any one of them. So I've covered all of them, so that hopefully you're able to grasp what's going on with each of them, and then you'll probably be asked about just temperature or just concentration. Okay, you don't tend to see more than one of them being asked in an exam question. All right.
do we have an example for acid reactions we could do?
Okay, so for an example for the acid reactions, if I think I've got one later on, but just to cover it while we're on this page, if I said hydrochloric acid, and let's add that to, which metal shall we? Yeah, magnesium's a good one. Magnesium. Then the salt, the naming of the salt, and I think that's the important skill here, is to use the name of the metal first. So we're going to write magnesium for the salt. And then the second part of the name of the salt comes from the acid that you've used. So because this is hydrochloric acid, it's going to produce a chloride salt. And then we're also going to get our hydrogen gas given off. Spelling's gone AWOL there. Hydrogen gas. If that example was sulfuric acid, then it would be sulfuric acid plus magnesium gives magnesium sulfate plus hydrogen gas. If it was nitric acid, then the example would be nitric acid plus magnesium produces magnesium nitrate plus hydrogen gas. Okay, so I think that brings us on to a little bit of atomic structure. And so you'll get a periodic table in the exam, and you should be able to look at those symbols and hopefully recognize most of them. You should know up to calcium, number 20. And when we look at that information, we can see that C, or carbon, has an atomic number of 6, and that tells us that it has 6 protons in the nucleus. If it's an atom, then we know that it's neutrally charged, so it has to have the same number of negative charges as positive charges. So because it's got 6 protons, it's going to have 6 electrons as well. And if we know the, I don't think you need to do it normally anymore, but if we knew the mass number, then we could work out by subtracting the atomic number from the mass number how many neutrons there were in the nucleus of carbon. Your work will tend to focus on the protons and electrons more. So the thing that we might need to then do from that is to pull out the information about the electrons and put that into an electron configuration. So I've given sodium and oxygen as examples, but if I add carbon to that, so I'll put carbon over here, we know that carbon had 6 protons and therefore 6 electrons. And that means that the 6 electrons are going to be written like this. We get 2 in the first shell, that's always the case, other than for hydrogen, which only has 1 electron. and then the next lot of electrons are going to be allocated to the second shell, and that can take up to 8. So because we know that carbon has 6 electrons, we've put 2 in the first shell, which means we have 4 left over, and so the electron configuration for carbon would be 2, 4. Sodium, we can see that sodium's got an atomic number of 11, so 11 protons, 11 electrons, and that's going to be arranged as 2, and then 8 in the second shell, and then if we add 2 and 8 together, that's 10, so there's only 1 left over when we take 10 away from 11, and so that last one's got to go in the outer shell of the third shell. Oxygen has an atomic number of 8, and so that's got 8 electrons when it's an atom as well, and they're arranged 2 and 6. Okay. I would definitely expect to see a question that asks you to discuss that in some capacity in the paper on Friday. Okay, so then from that kind of basic atomic structure information, we're going to go into how do atoms form ions. So we've outlined sodium and oxygen. We're going to use those throughout this example. And so here is a diagram showing our sodium with 2 in the first shell, 8 electrons represented by X's in the second shell, and 1 electron in the outermost or valence shell. Oxygen, by comparison, has its 2 in the first shell, and then it's got 6 in its outermost shell. Okay, when we are describing or explaining how atoms form ions, we're really focusing on these outermost electrons, and so that's why I've highlighted them in red here. And so what's going to happen is that sodium is going to, in order to achieve a full outer shell, it's either going to gain enough to make a full shell, so it would have to gain 7 to get 8 in this outermost shell, or it can lose 1. And because losing 1 is easier, sodium tends to lose 1 electron. And so this little electron here would disappear, and sodium becomes an ion. It's now charged because it has 11 protons, but it's only got 10 electrons now. So 11 positive charges and 10 negative charges means that it has a single positive charge on it. And I would normally show that as being a little plus up to the top right of it. Oxygen, by comparison, is going to gain, in order to fill its outer shell, it's going to gain 2 electrons. Okay, so I'm going to take one of the electrons here and a second electron in order to fill its outer shell. Now, once it's got a full outer shell, it's still got 6 protons in the nucleus. It's now got, my bad. It's got 8 protons in the nucleus, and it's now got 10 electrons, which means it's got 2 extra negative charges. So oxygen becomes 2 negative. And what we've done there is we've explained how those atoms form ions, so how they go from being neutrally charged to being charged. And in terms of what's really happening there, takes us into the next step of how ions bond together. And so in this case, we had 1 electron from sodium, and it had come over to the oxygen, which means that in this case, we're still missing an electron from oxygen. So what has to happen is that we get a second sodium ion. So here's our second sodium ion up here, and because it's got that excess electron in its outer shell as well, I need to ungroup that. Then it's going to lose that outermost electron, and that is where the second electron comes from for the oxygen ion. And so in doing that, we've now got 2 positively charged sodium ions, and we've got 1 negatively charged oxygen ion. or oxide ion, I should say. And so we now have a negatively charged ion and positively charged ions, and so there is a force of attraction between those. So that force of attraction bonds those ions together. So they create, in forming ions, they created either positive for sodium or negative charge for the oxygen, and then in becoming positively and negative charged, they're attracted together. And that's how ions form compounds. And so because we need 2 sodium ions to balance the charges with 1 oxide ion, the formula would be Na2O. And we don't need to write any number after the oxygen because it's just a single oxide ion. Okay. I would expect to see a question in which you are required to explain that ratio, how we get a ratio. And the question would give you, different atoms or different ions to work with, and then you would have to use those in your exam, in your explanation to arrive at a ratio between them. Okay. Some of you may have been taught the swap and drop, and you can use the swap and drop to identify that formula as well. But I like to think about it in terms of balancing charges, and so because I know that I need 2 sodiums in order to balance the charge with 1 oxygen, then that's going to give me a 2-to-1 ratio of sodium to oxygen. Okay, so in terms of do we need to draw diagrams in the exam? No, you don't, but it's actually really useful in terms of explaining what you're doing. I think it's useful for you, and it's sometimes useful for the examiner. So I would recommend drawing the diagrams. Often with the electron shell diagrams, you can get the credit either by writing the 2, 8, 1, or by drawing those diagrams. Okay, and if it helps you to explain it, then draw the diagrams. Once the ions have bonded together to form a compound, that compound is now neutral. And it's in a relatively stable state, I guess. Okay. So this is an explain the ratio question. That's what we've just gone through. So I'm not going to dwell on the answer to this one. I'm just going to reveal the answer, give you guys a chance to have a quick look over that. And again, you'll be able to go through this video on your own time and pause it, take a bit more time if you need it. So in this one, you've had magnesium fluoride given to you. You've been told the formula. You might also have to work out the formula from, just from magnesium fluoride. You're asked to explain the ratio of the ions and how that's linked to charge on the ions. So we would go through the same steps that we just did. We've got the electron structure for magnesium and fluorine. We then describe what's going to happen to those outermost electron shells, the 2 electrons from magnesium being lost, and the fluorine gaining 1 electron to form the fluoride ion. And then that creates that negative and positive charge which bonds those ions together. And because the fluorine is going to gain 2 electrons, because the magnesium loses 2 electrons, in order to balance that charge, we're going to need 2 fluoride ions, and so that gives us the formula MgF2. All right, we've only got about 10 minutes left for acids and bases, and I think one of the most important skills for acids and bases is being able to write word chemical and balanced chemical equations. So I'm going to move on to that fairly rapidly. I'll just give you another moment to have a look through that. It's a good question there about the the valence shells there in terms of the periodic table. If you did have to answer that, you would go down and identify that lithium with 3 electrons, i.e. 2, 1, sodium with 11, 2, 8, 1, Potassium, 2, 8, 8, 1, all of them have one outermost or valence electron, and so that's why they're all in group 1. And similarly, if you go through, you'll find beryllium, magnesium, and calcium all have 2 electrons in their outermost shell, and so they're in group 2. Whereas if we go over to the other side of the table, the noble gases here all have stable outer shells, full outer shells, so we don't tend to see them in any reactions. Whereas when we get into group 17, as listed on this periodic table, then that's fluorine, chlorine, and bromine all have 7 electrons in their outer shell, so they're all in that group because they've got the same number of electrons in their valence shell. Good question, thank you for asking. Alright, so writing and balancing chemical equations. The table of ions that you're going to get in the exam is really useful here. You're probably going to need to refer back to that. I would always refer back to it just to double check, even if I thought I knew all of the charges. And so in this case, we're being asked to discuss how the student would make magnesium chloride salt from hydrochloric acid and magnesium oxide. That's going to be a neutralization reaction between a metal oxide and an acid, so that's the state, what type of reaction occurs. State implies it's really just a very simple, probably one word answer or short phrase, and that's just an achieved level. Depending on how well you answer the second bullet point will determine whether you get achieved credit, merit credit, or excellence credit. So we start off by writing a word equation, and so we would take hydrochloric acid and magnesium oxide, the reactants, because the student's making magnesium chloride, that's the product, so it's going to be on the right hand side of the equation. So here we've got hydrochloric acid plus magnesium oxide, and that produces magnesium chloride. So we take the name for the salt from the metal, and the chloride comes from hydrochloric acid. And we don't really need to understand that for this question because we're told it's going to be magnesium chloride salt. And then because it's an acid plus a metal oxide, that produces a salt plus water. And so that, you'll remember that we talked earlier on about the need to know. You need to know these 4 reaction types. 4 reaction types. I forgot that was my example there. So the 4 reaction types, what is produced in each of those. So acid plus metal oxide is salt plus water, and that's what we're seeing down here. Acid, metal oxide, salt, and water. So then we would use that idea of balancing charges to write our formula. Well, we hopefully don't need to for hydrochloric acid. We should have that memorized, HCl. Magnesium oxide, we would look at the table of ions. We would see that magnesium has a charge 2+, and oxide has a charge 2-. So in order for those charges to balance, we just need 1 magnesium and 1 oxygen, and so we would expect the formula to be MgO. Magnesium chloride, again Mg2+, and the chloride ion is Cl-, so we're going to need 2 chlorines with a single negative charge each to balance out the single magnesium with a 2+ charge. And so that means the formula for magnesium chloride would be MgCl2. And water we should know is H2O. Okay, so that gives us, there's our formula. So from what I've just said, we would have everything except for the 2. So the word equation, if you can get that far, that's normally achieved level. If you can get the chemical formulas, so if you just got to HCl plus MgO, gives MgCl2 plus H2O, that would be merit. The excellence comes from balancing the equation, and so to do that I would go through both sides of the equation. I'm actually going to take that 2 out there because it makes more sense if I'm talking through it as I go. I'm going to count up the number of each element on the reactant side and compare that to the product side. So I've got 1 hydrogen on the reactant side and I've got 2 on the product side. I can't mess around with the numbers after anything or the numbers between any formulas. All I can do is put a big number at the front. So in order to get, because there's 2 hydrogens over here, I'm going to need 2 lots of hydrogen from the hydrochloric acid, which means I need 2 whole molecules of hydrochloric acid. So I'll put a big 2 in the front of that. Now the hydrogens are balanced, so then I check through the rest of it. Chlorine, well, I've now got 2 of those because of the big number 2 in front, and I've got 2 chlorines on this side as well, on the product side. I've got 1 magnesium here, and I've got 1 magnesium here, and I've got 1 oxygen here and 1 oxygen here. So that is now balanced. I would recommend that once you've made any changes to your equation, like by putting numbers in front of things, you go through one last time at the end and just double check from the beginning to make sure that everything is correctly balanced.
I've got a question about, do we need to know about electrospheric interactions, and also a lot of people have been talking about isotopes in the periodic table. Do they need to know?
So good questions there. I see there's some questions on isotopes. I think isotopes were actually removed from the standard a number of years ago. So while we often still teach it, it's not normally assessed. So you don't, from the best of my knowledge, and now I want to check the standard to make sure, you don't need to know about isotopes for the exam. There was another question in there about electrostatic attraction. That's just the name for this. You know, where we've got the positively charged ions and the negatively charged ions. Electrostatic attraction is just the force between those, and that's all you need to know about it, is that you know, a positive ion will be attracted by a negative ion, and vice versa. All right, so here's another little bit of practice, just I think to finish us off with. Oh no, actually I might, looking at the time, I might skip past this one. This is just another example of the word equation and balanced symbol equation. Again, no brain too small is really good for that. You can find something called the Balancing Equations 5 a Day on there, and that's got all the answers. So if you're struggling with this concept, practice them through any means necessary, but no brain too small is a really good one. I will just reveal these answers so that if you're watching back on the video, you'll be able to see what the correct answer was for each of these. And there's another example, I think. So I'll pop those down there just so if you're watching back on the video, you can practice those for yourself and see if you can get the right answer. All right, that brings us to the last little bit, I think, of acids and bases that I wanted to cover for you. And that's where we have to describe what's going on in a neutralization reaction with regard to the concentration of hydrogen ions and hydroxide ions. So you often have to relate this to perhaps color in universal indicator and pH, remembering that you know, the pH scale for our purposes normally is considered to run from 1 to 14. It's actually a bit more complex than that, but that works for level 1, with 7 being neutral, and anything below 7 being acidic, and anything above 7 being alkali. So if there are an even ratio of the hydrogen and hydroxide ions, and by that I mean if there's 3 hydrogen ions and 3 hydroxide ions, that's an even ratio, okay, 3 to 3. Then the solution is neutral, pH 7. And so in this diagram down here, we can see in the solution there's a whole load of OH's floating around and a whole load of H's floating around, and at the moment that's not actually neutral because we've got 6 hydrogen ions and we've got 4 hydroxide ions. So what we need to do in order to make that neutral is to, do I have that grouped or is it just a static image? Yeah. So I think I've just got that as a static image from a diagram I'd used previously. So this is a little bit misleading, I apologize for that. This would need to have 4 hydroxides and 4 hydrogen ions in order to be pH 7, or it would need to have an extra 2 hydroxide ions to make it 6 and 6. So I might just note that on there. If the pH is greater than 7, then that's an alkali, and so what we would expect to see there is a higher ratio of hydroxide ions compared to hydrogen ions. So fewer hydrogen ions with more hydroxide ions, and that means that the pH is over 7 and it's alkali. If the pH is less than 7, it means that there are more hydrogen ions than hydroxide ions, and so in this case we can see that there's about 9 hydrogen ions and only 4 hydroxide ions, so we know that the pH is less than 7. So in your explanation there, and again this is a really common question, being able to describe what's going on in a solution with reference to the hydrogen ions and the hydroxide ions, and relating that to pH and possibly to colour using universal indicator or litmus paper.
So we've got 8 to 11 or 8 to 10, does that matter? I think that's for weak bases. And also, do you have any of the mammals' chem reactions at
No, sorry, I don't have any videos on the other standards. It's just the mechanics, the acids and bases, and genetics. I'm not sure if you might find some for mammals on YouTube. It'd be worth a search. For a question on neutralization, do we need to go from the pH of 8 to 11 or 8 to 10? You might have to. It might be useful to be able to go through like 1 to 3, 4 to 6, 7, and then 8 to 10, and then 11 to 14. And in each of those cases, it's really just stating what's happening to the concentration of the hydrogen ions. So for a very low pH, there's way more hydrogen ions than there are hydroxide ions. And then as we get into a weaker acid, there's still more hydrogen ions than hydroxide ions, but the proportion is lower. So there are if we said that the ratio was 7 hydrogen ions for every 1 hydroxide ion at pH 2, then at pH 5 it might be 3 hydrogen ions to 1 hydroxide ion, and then for pH 7 it would be a 1-to-1 ratio. pH above 7, so maybe in the 8 to 10 bracket, we might be saying that there are slightly more hydroxide ions. There are slightly more hydroxide ions than hydrogen ions if the pH is 8 to 10, whereas if we went to pH 11 to 14, there are way more hydroxide ions compared to hydrogen ions. Okay, so I think it's gonna be time for acids and bases. Yeah, question about spectator ions. To the best of my knowledge, you don't need to deal with spectator ions in the level 1 acids and bases paper. You could go and double check the standard for that, but yeah, I'm pretty sure that's the case. We don't need to worry about spectator ions. Okay, so I'm going to move on now to mechanics, the last of our 3 papers. Okay, and so for mechanics, I always recommend the guess method. Hopefully by now a number of you will have seen this. Maybe your teacher taught you it, maybe you found it online or seen it on one of my videos. This is a really good approach to help you, even if you feel you've got no idea about a calculation question in a mechanics paper, you should be able to follow these steps, and in doing so you should be able to get to an answer. And so even if it's not entirely correct, you might be able to get some credit for it. So the guess method is an acronym where each letter stands for a word. So G is given, and you just write down what you've been given in the question, like M equals 120 kilograms. U is the unknown. What value have you been asked to find? Again, that really just comes from reading the question. So it might be that velocity is an unknown, so V equals question mark. And when I use the term velocity there at level 1, that's interchangeable with the word speed. E is then an equation, and so you'll have this at the front of the booklet, and you should be able to then look at that and go through and tick each of the letters that you've got written down as being given or being unknown, and then that should identify one of those equations that you can use. And it might be for an excellence question that you have to use two of them, so you have to work out something from the first equation and then put that into the second equation. That's quite a standard excellence answer. So once we've identified the equation, we write it down in our answer, we put the numbers in, that's the substitute, and then we solve. So we pop those numbers in our calculator, being careful, watch out for things like fractions and squared. Those tend to be issues that cause students lots of troubles in the exam. So EK equals half mv squared, you've got to make sure that you square that velocity if you're working out kinetic energy. You know, be careful with the keys you're pressing on your calculator. You know, you might be wise to use brackets depending on how you're approaching your calculation. Make sure you've got the right units at the end of your answer. You know, so for example, speed is normally measured in meters per second, so you always write the units that you've measured it in. Significant figures don't play such a key role in level 1 science, so I would probably just put my answers to 2 or 3 significant figures. From memory, you're not going to be marked down for that. And do a common sense check. Like, look at your answer and think, does that number make sense? And that's a really good safety check for you. So that if you've made a mistake, sometimes that common sense check is going to highlight that, and then you're going to be able to go back through your working and find out where you've, you know, made a mistake. Okay.
Is there a recommendation for a calculator?
Oh, look, any calculator will do. I think most of the schools I've worked in and students that I've known have one of these Casio calculators. Like you could get by with a very, very basic calculator. It probably helps you to have something that's got a squared function on it, but you could achieve that just by multiplying the number by itself. I think yeah, any calculator should be sufficient. So one of the biggest questions from you guys in the form around mechanics was to do with force and power. Sorry, I should say work done and power. And so what I've got here is a question about force with relation to work being done. Work being done is just another term for energy, so don't be confused by that. Here we've got a force of 100 newtons and a distance of 150 meters. So if the question said to me, you know, how much work is done if 100 Newton force is applied to an object over a distance of 150 meters, and I should probably say like a net force of 100 newtons or an unbalanced force of 100 newtons. No, sorry, that's not right. Yeah. An applied force, I suppose, might be a better term to use there. So if a force is applied over a distance, then how much work is done. And so we've written down what we've been asked to, what we've been given. The work done would be what we're trying to find. And so then we would flick back to our equations and we'd be looking for F, D, and W. And we find this equation has all 3 of them. So this is the equation we're going to use. So we write down our equation and then substitute the numbers in. So there's our equation, pop the numbers in, and then we write our final answer with units. So work done, as I said, is a form of energy. 100 times 150 is 15,000, and that's joules. Okay, you can use the symbol or write the word. Now, what we might then do, because power came out really strongly in that form, if we take, if we said that the time was 5 seconds, then the question might be, what is the power in this situation? So I put power as my question mark. And because we've already got the work done or the energy, We've got time and we're being asked to find power. We then flick back to our equations. There we go, power, work done, and time. And so we would use this equation. We couldn't use this equation, that's power, pressure, force, and area, because we don't have all of those quantities. So P equals W over T, power equals work done divided by time. We go back over to our question and we write down P equals W divided by T. Then I would substitute in my numbers, 15,000 divided by 5, and that's going to give us 3,000 watts. Okay, so watt being the unit for power. All right, and then down the bottom I've put what is the speed of a 20 kilogram object after 150 meters. So if we'd applied that force to it, so we've got we've got the work done there, the 15,000 joules. So the idea is that that amount of energy, due to the law of conservation of energy, would be our EK value, and then that's half mv squared. So we can rearrange that. I haven't gone through the rearrangement here. I might do that if we've got time a little bit later. And so we would be doing the square root of 2 lots of EK. So 2 lots of EK would now be 30,000. And we would then divide that by the mass. And so we've got the square root of 30,000 divided by a mass of, what did we say, 20 kilograms. Okay, and that comes out as being 38.7 meters per second. So that's just another example of a direction you might go in with a question relating to force or work done. So that's our basics about work and power. Power is just the rate of transfer of energy. It's measured in watts or joules per second. And that's our formula. I've used E in this equation just because it's energy, but in the resource booklet it's listed as W for work done. Remember, they're just the same thing. Okay, for excellence, you will have to explain how two physical quantities like work and power relate to one another. And so what's happened here is it's given us a situation. We've got, if we had 2 people on 2 different horses, with this question we've got 1 horse, and if we made the person on the horse heavier, then what effect would that have on speed and acceleration over a race? And this is just part of an exam question, so hopefully it hasn't confused you in that you're not seeing the A and the B part. I think we're going to have a look at those in a little bit. I've just put these ahead because work and power was more of a priority on the form. It says calculations are not required. You can use calculations to support your answer. If you feel more comfortable putting numbers in and seeing what comes out, that can support your answer. But what you definitely want to do is to find equations that relate these things together, and then make sure that you talk about every quantity in those equations. So in this case, we would say that a heavier or larger mass rider with the same speed and acceleration would require a larger net force to maintain the same acceleration. So I'd probably be using the equation F equals ma in there. And I would actually write the equation that I'm using to support my answer, and I would tick off each of those letters as I've talked about it, because often you get credit for saying that something stays the same. So if something stays constant, you've got to explicitly state that. As the force applied must be larger, the work done will be greater over the same distance. So that's using that formula, work done equals force times distance. And then if our work done or energy has increased, then because we've got that formula, power equals work done divided by time, and the time remains unchanged. That's an example of what I was referring to when I said you've got to, if something stays the same, you've got to explicitly state that. if the work done increases, then the power must also be greater. So hopefully that gives you an example of how you might frame your answer to a question.
Appreciate your questions.
If you were to write the equation and not the answer, would you still get a mark? Not without any, not normally without any additional steps. If it's a calculation though, and you've got the equation, you should be able to put in the numbers and get to a a final answer. If the use of the equations was in an explanation, then you would need to refer to, you'd need to describe what was going on in that equation in order to get some credit. And if you could, depending on how well you could do that, might determine whether you got some achieved or some merit or some excellence credit. Okay, so sticking with energy, there's a good chance that you'll see a question that relates an object falling or sliding down a ramp where the height changes and there's something about its velocity. So either it tells you how high up the object's dropped from and is asking you to work out its velocity at the bottom, or it might give you the velocity and ask you to work out how high that object was dropped from. So in this case, if we had a skateboarder who started at the top of a 6-meter high ramp, then if we're ignoring friction, what would their speed be at the bottom? And so this uses the principle of the law of conservation of energy, and you often get credit just for stating the law of conservation of energy, or EP, potential energy, equals EK, kinetic energy. So it's really important to remember to write that at the start of this question. What that means is that because EP is mgh, that's mass times gravitational field strength times a change in height, and EK is half mv squared, then we can write those down as being equivalent. What I'm going to do here is I'm going to lead you through some rearrangement of formulas, and then I'm going to come out with a little shortcut formula for you to use in the exam if you want to. So we can, because EP is mgh, EK is half mv squared, we can write those next to each other with a little equal sign between them. I don't like fractions. I find them hard to work with, so I'm going to multiply both sides of my equation by 2, and that gives me 2 mgh equals mv squared. So I've doubled both sides of the equation, and that's given me 2 mgh, and that's got rid of the half. So instead of having half mv squared, I've now got mv squared. Because we've got m on both sides of the equation, we can divide by m to get rid of the m's. And that gives us 2gh is equal to v squared. And then I can square root both sides. Now don't worry too much if these steps are kind of going over your head. Ultimately, you might just need to know the end step here. But we can square root both sides, and that gives us the square root of 2gh is equal to v. So square rooting v squared gets us back to v, and square rooting 2gh gives us the square root of 2gh. Now, if we know that we're on Earth, then g is 10, as stated in the resource booklet, or the little bit at the start of your exam. And so we can actually simplify this formula even further, and we can get down to a stage where we've got the square root of 20h, because 2 times 10 is 20. Square root of 20h equals the velocity. So in the situation of a 6-meter high ramp, whether that's a drop or sliding down a ramp, the change in height would be 6 meters, and so we would do the square root of 20 times 6, and 20 times 6 is 120, so we would do the square root of 120, and that would give us our velocity. And if you try that on your calculators, then that should come out as about 11 meters per second. Okay. And because we've got this equation here, 2gh equals v squared, we can turn that equation around the other way. So if we weren't interested in v, but we were interested in height, then we could also work an equation to be, where shall I write it? Pop it in over here. So if we rearrange that equation, then we get h is equal to v squared, oops, divided by 2, divided by 2g, and that is a squared. There we go. So what that means is that if an object's fallen and we know its velocity, then we can work out how high it's fallen from to reach that velocity. So maybe if we try that with, if you went skydiving and you know, after jumping out of a plane you had fallen say 200 meters, then no, sorry, I've given that the wrong way around, haven't I? If I said that you had reached terminal velocity, which I think is about 53 meters per second, then what height might you have fallen from? Then what we would do is we would put 53 meters per second into our equation, 53 squared, divided by 2g, and if we're on Earth, that g is going to be 10, so that's 20. And that gives us a change in height of 140 meters. Okay, and so that's just two equations there that hopefully make life a bit easier for you. But I would always write down that you're getting that from the conservation of energy, which is EP is equal to EK. You'll often have to explain something around that, like it'll say, well, in this case, the skateboarder slid down a 6-meter high ramp and he reached this velocity of 11 meters per second. How would that compare to reality? And in reality, we've got friction, and friction means that some of the energy is being lost. So it's not being converted from gravitational potential into completely kinetic energy. Some of that kinetic energy is being lost as heat or sound or light. And so there's less kinetic energy, and if there's less kinetic energy, then it means that the object's velocity, or the skateboarder's velocity, wouldn't be quite as high as 11 meters per second. All right. I've skipped past distance time graphs because I can't remember when the last time we saw a distance time graph in the exam was. We tend to see the speed time graphs more. And there's a lot more that you can be asked about with the speed time graphs. So for a speed time graph, we should know that we can read off the speed at any given time. So if we look at this red line here, I could see that in this horizontal red section, the speed is 9 meters per second, halfway between 8 and 10, and it's continued at that speed from 3 seconds to 14 seconds, so it's continued for 11 seconds at 9 meters per second. The steepness of the line indicates the change in speed, so that's the acceleration. A steeper line indicates a greater acceleration, and that can be speeding up or slowing down. If the line's horizontal, it means that the speed is constant. The other thing that you can be asked about with a speed time graph is the distance traveled. And the distance traveled is given by the area under the speed time graph. So to get that, we would split the shape of the graph up into triangles and rectangles, and then we would use that information, the area of each triangle and rectangle added together to give us the total distance traveled. So let's have a little look at what those look like. Acceleration equals a change in speed over time. So for bird A, if we were to look at, I think this section here, we've got the blue line as bird A. We've gone from 0 to 10 meters per second, and that's happened over a time of 4 seconds. So in this section of the graph, it's 10 divided by 4, and that would give us 10 divided by 4 would be 2.5 meters per second meters per second squared for its acceleration. Now I think, so the question here relates to the first 3 seconds of the object's motion. So while the example I just gave was using the 10 meters per second and 4 seconds, this question over here relates to the blue and red points at the 3 second mark. So for the red line, bird B, we've got over 3 seconds, we've got a change in speed of 0 to 9. So 9 meters per second divided by a change in time of 3, which gives us 3 meters per second squared. If we compare that to the blue line, we can see the red line steeper, so we should know that the acceleration is greater. Both bird A and B then have these horizontal sections, and that means the acceleration is 0 because the speed is not changing. And then we could also work out the acceleration, which you might also call the deceleration, over this section. If we looked at the bird A as an example, then we're going from 10 to 0 meters per second, and so the change in speed is effectively negative 10, going from 10 to 0, and then the change in time is between 14 and 16 seconds, so it's 2 seconds. So we would end up with an acceleration of negative 5 meters per second squared, which you could just say is a deceleration of 5 meters per second squared. For the distance traveled, I think the example I've given here has us working out the distance traveled by bird A in 16 seconds. And so we split it up into a triangle. This dotted line gives us the first triangle, and then a rectangle between 3 and 14 seconds, and then another triangle between 14 and 16 seconds. Sorry, that was bird A, I was looking at bird B. For bird A, the first triangle is between 0 and 4 seconds. Then we've got that rectangle, and then we've got the triangle formed between 14 and 16 seconds. So our first triangle is 4 seconds times 10 meters per second, and it's a triangle, so we half that to give us 20 meters. The rectangle is between 4 and 14 seconds, so that's 10 seconds, and the speed is 10 meters per second, so it's 10 times 10. It's a rectangle, so we don't half it, and so that's 100 meters. And then the last triangle is 2 seconds, again times 10, but it's a triangle, so we half it, so that's 10, and so adding those 3 distance values together gives us a total distance of 130 meters. And we might be asked to compare those. So in this case it says how much further did bird A travel in the same time. So if you just said 130 meters, you haven't actually answered the question. You have to show the difference between those distances to fully answer the question. I'll leave that up for a moment and just check any questions in chat.
About units for getting them correct. But it's not questions, more like just chatting.
All right. Well, we're nearly there, I think. Let's see what I've got ready on the next slide. Pressure, force, and area coming up here, I think, with mass and weight. So quite a common question is to define the difference between mass and weight. Mass is a measurement of the amount of matter in an object, and it's measured in kilograms, and weight is a force produced by the interaction of mass and a gravitational field, or gravity. And so weight is a force that acts downwards, measured in newtons, and mass is a measure of the amount of matter in an object that's measured in kilograms. And so here we've been asked to include a calculation for weight, and so using the guess method, we've got mass, we've got G for gravitational field strength, Our unknown is the weight force. Our equation then is F equals mg, and that's force is equal to mass times g, which is 10, giving us a weight force of 99 newtons. Similarly, we might have calculate the weight of this time it's a kerru, a mass of 630 grams. I've shown you this one because you have to convert the grams into kilograms. So when we write our guess method, if we see something in units that we wouldn't normally use in the calculation, like grams, then we're going to change that into kilograms. And so we should know that there's 1,000 grams in a kilogram, so we can divide 630 by 1,000 to get that into kilograms. G is 10, and then weight force again, write the formula, substitute the numbers in, and solve to get 6.3 newtons. Always remembering to check that you've got the right units for the question. So it's good to go back and reread the question and just double check that your units match that. Okay, so then I think that gives us just enough time to finish with pressure, force, and area. So with area, I might go into a new slide and just draw this one out. What you might have to do is to work out the area of an object. So if we had, let's say, a table leg, and this is like a cross-sectional area of one of the legs, then it might be, let's say, might be about 3 centimeters on one side, and it might be a little bit more on the other side. Let's say that is 4 centimeters down here. Okay. In order to work out area, we might have to multiply those together. I can't think of the last time I saw us having to do that, but it's probably a good skill to have just in case. And so this table leg would have an area for one table leg of 4 times 3 centimeters, giving us a result of 12 square centimeters. I don't like working in centimeters and it doesn't fit with the equation, so what we're going to do is we're going to convert that into meters. And so we know that 3 centimeters, there's 100 centimeters in a meter, so we can divide that 3 by 100 to give us 0.03 meters, and the 4 centimeters is 0.04 meters. Now when we multiply these together, we're going to get the right units in meters squared that we can use in our equation. And so 0.03 times 0.04 should come out at, what's that going to be, 0.0004. I'm just going to double check that on my calculator, which is always good advice for the exam. Yeah, I think I've got that right. Meters squared. Okay, so that is our area. Now, if we were told that the mass of the table was 35 kilograms, then we would need the force in order to work out pressure ultimately. So the mass allows us to use the equation we saw before, F equals mg, and so we can identify the force as equal to 35 times 10, which would give us a force of 350 newtons. All right. So then we can start thinking about the pressure exerted by the table on the floor. And so we know from our equations at the front that pressure is equal to force divided by area. or P equals F over A for short. We can see that the force is 350 newtons and the area is 0.0004 meters squared. But there's a little trap there because, you know, a table normally has 4 legs, for example. And so if our table has 4 legs, then our area needs to be multiplied by 4 in order to get the correct pressure. Oh, sorry. Yeah, thank you. That should have been 1, 2. Senior moment there. Completely missed that, so thank you for pointing it out. Let me just check I've got that right. 1.2 times 10 to the negative 3. Nope, I've got 1 extra 0 in there. Okay, I think that's correct. If you guys would like to double check for me and let me know in chat if I've made a mistake with that. So then the area that we're working out, because there's 4 table legs, we would then multiply that by 4. And so that would give us 0.0048 meters squared, I think. And so then our equation becomes P, pressure equals force of 350 newtons divided by 0.0048. And then we'll just check that on our calculators, substitute the numbers in and solve to give us, I have got 72 or 73,000 to 2 significant figures. So 73,000, and that's measured in pascals for pressure. The alternative units for pressure, if you forget that it's pascals, you can see in the equation that we've divided force, which is measured in newtons, by area, which is measured in meters squared. And so we could also say that that is, or newtons per meter squared.
Okay. An excellence question.
Normally this level of working would be an excellence question. So a good clue for an excellence question in the mechanics paper is if you've had to use 2 different equations to arrive at your answer. So in this case, I would suggest that we'd use the force, the weight force. We had to work that out, and then we had to put that into the pressure formula to get our final answer. So generally, if you use 2 equations, then it's likely to be an excellence level answer. All right.
And force diagrams.
Okay, and there was, I think I've probably got just enough time to rush through a couple of force diagrams. So here's one that I like to use, the drone. If the object's at rest on the ground, then you know that the speed is constant. In this case, it's stationary, but that just means a speed of 0, doesn't it? And so if the speed is constant, then the forces acting on it are balanced as per Newton's first law of motion. So the support force going up is the same size as the weight force going down. I would, if you draw force diagrams in the exam, I would take a ruler in with you and make sure that if they're meant to be the same size arrows, you draw them as the same size. And I would use a ruler for any arrows. Remember to put your arrowheads on and to label your forces. So in this case, the drone has a support force because it's resting on the ground, and it's got a weight force acting down. And the magnitude, the size of each of those arrows is the same. So there is 0 net force. If it was rising at a steady 5 meters per second, then it's got a weight force acting down. And it's got, because it's bumping into air molecules as it's rising, there's a frictional force acting on it as well. And so the combined weight and friction force would equal the total lift force. Because it's a steady 5 meters per second, we know that that means that there is 0 net force. So however much upwards force must be the same as the total downwards force. I wouldn't expect you to have to. Oh, you don't often see the combination of different forces in the free body diagram for one direction there. So I don't recall often seeing you having to combine weight and friction to produce the same size arrow as the lift arrow, for example. But being better prepared for it hopefully helps you understand it and apply it to any situation. If the object's accelerating, then from Newton's second law, we know that it must have an unbalanced force acting on it, which means that one of the arrows is going to be bigger than the others combined. So in this case, I've got one arrow going upwards from lift, and I've got two arrows going downwards. I've got weight and friction again. And so the combined weight and friction acting downwards is smaller than the lift force, which means that there's a net or unbalanced force upwards, which means the object is accelerating upwards. You should only have to deal with this in one dimension, so vertically or horizontally. You won't be asked to normally combine them in multiple directions. And you could apply that to a parachute situation as well. If a person jumps out of a plane, the instant they jump out of a plane, their weight force is bigger than their friction force, so they're accelerating. A moment later, they are now faster, so they're bumping into more air molecules, so their friction force is increased, but they've still got a net force downwards, which means they're still accelerating or getting faster. And then at this stage, what I've labeled arbitrarily as time equals 3, the friction force equals the weight force, and so the forces are balanced and the person has reached a constant speed or terminal velocity. They are no longer accelerating. If they then pull the parachute, we get a much bigger friction force and the same weight force, remembering their weight force can't have changed, and so the much bigger frictional force upwards means that they are now decelerating, so they are slowing down as they fall.
Thank you. I think it's past 8.
All right, I think that's